So now that we've talked about cellular reproduction in general, we can take a closer look at mitosis, which is the process that cells use to divide and make identical copies of themselves. So this is a key part of the cell cycle, and it's going to happen in all of our body cells, and our body cells are referred to as somatic cells. So before the cell divides, it's going to duplicate its chromosomes. The duplicated chromosomes are made up of two sister chromatids that are joined at a central point called the centromere. So here's our original chromosome. We're going to undergo replication, at which point we're going to produce two sister chromatids shown to you here that are joined at a central point called the centromere. And each sister chromatid is going to contain an identical copy of DNA. So when the cell eventually divides, each new cell will get one of these chromatids. So that's what it's showing you here. So before mitosis can even begin, the cell is going to spend most of its time in a phase called interphase. And this is the longest part of the cell cycle, and it's when the cell is doing its everyday work. So growing, making proteins, and preparing to divide if needed. So how long a cell stays in interphase is really going to depend on what type of cell it is. Interphase is divided into 3 different stages. The first one's going to be the G1 phase, or GAP1. And this is the stage before DNA replication. So here is where the cell is going to be growing, doubling its organelles, and gathering its materials that are needed for DNA synthesis. It's also at this point that the cell is going to decide whether or not to divide. So some cells are actually going to exit the cycle and enter into a different phase called the G0 or G0 phase. And if they enter into this phase, the cells will not divide, and sometimes cells will stay in this phase indefinitely. We also have the S phase, or the synthesis phase, and this is where DNA replication is actually happening. So by the end of this stage, each chromosome has turned into 2 sister chromatids that are joined at a centromere. So we just saw that on the previous slide. The G2 phase, or the GAP2, is going to come after DNA synthesis, and it's going to continue right up until mitosis begins, and the cell's making proteins and other components that are needed for cellular division. So once interphase is complete, the cell is going to enter into the M phase, or the mitotic phase, which is where the actual division is going to happen. So the M phase includes 2 big parts. We have mitosis, which is the division of the nucleus, and then we have cytokinesis, which is the division of the cytoplasm. And together, they're going to result in 2 genetically identical daughter cells. So during mitosis, the cell's duplicated DNA is going to be carefully separated so that each daughter cell gets a complete set of chromosomes. The sister chromatids that were copied during the S phase are going to be split apart, and they're going to become individual daughter chromosomes. So that process is shown to you over here. So it's important to note that the daughter nuclei produced at the end of mitosis are genetically identical, not only to each other, but also to the original parent nucleus. So in animals, chromosomes always come in pairs. 1/2 comes from each parent, and diagrams sometimes color code these to show which one they came from, either one that came from your mom or one that came from your dad. So to separate chromosomes, the cells use a special structure called the spindle. So most eukaryotic cells use this mechanism to pull chromatids apart. And the spindle is made up of microtubules, which are just part of the cell cytoskeleton, which we've talked about previously. And these microtubules grow out from the centrosomes, which act as the main organizing centers for the spindle. The spindle fibers are going to attach to the chromosomes and help move them during mitosis. So there's 4 main phases to mitosis, and the acronym for this here is PMAT, and it's going to put them in order. So we'll start with the P, which is prophase, and this is where the chromosomes are going to condense and become visible under a microscope. Each one is made up of 2 sister chromatids that are joined at a centromere. Then we have metaphase, which is our M in PMAT. The chromosomes are going to line up in the middle of the cell along what's called the equatorial plate, or the equator, or the middle of the cell for metaphase. And this alignment is really important for ensuring that everything is divided evenly. The A in our PMAT is going to be anaphase, and this is when our sister chromatids are going to be pulled apart and moved to opposite poles of the cell. So once they're separated, each chromatid is going to be considered a full-fledged chromosome. Then we have our T, which is telophase, and that's where our nuclear envelope is going to form around each set of chromosomes, and the cytoplasm is going to split into two during cytokinesis. So at this point in our microscope, you can clearly see two distinct cells that are forming.